wants to know. I've had a few questions about the assignment, and as a lot of you have found out, the due dates are pretty flexible. So if somebody, I know they were due at the start of class, if somebody is learning new material that helps them do better on the assignment, you can go back and resubmit that assignment. I think you're allowed to submit it twice. If not, let me know. The assignments are really a way for me to give you material that will mimic what's going to show up on the exam. And the exams, you don't get really a second chance at that. So I want you to be able to work through the assignment problems and hopefully get them correct. So looks like a lot of people were kind of comfortable with the starting content, and then questions 8, 9, and 10 seem to be where a lot of people were hung up. I think part of that might have to do with a lot of it hinges on a very basic concept here, which is the idea of the coding strand. So I thought, I want to avoid giving you the answer to this, but I thought I could maybe give you a similar problem from a different gene and see if that helps things out a little bit more. So are there any, I don't know. I was going to ask if there are any questions, but they might just be, hey, can you continue with that thing you were doing? All right, so maybe I will, before we dive in on this, just kind of review the framework for what we would expect to find in a gene. The display is different today, and it's now forcing everything into this teeny tiny space. I don't know what to do with. Move. Squish. I need a better drawing pad. Let me see. View. All right. I guess we'll make do with what we got here. So if we start in with a piece of DNA that is a gene in a eukaryote, when we encounter genomic DNA is double-stranded. And so it has this code on there that in a gene has the code for all the steps needed to process, like to make the RNA, to process the RNA, to translate the RNA. All that code is contained in the gene. And the gene could be running in either direction. So we could have, there's nothing that says that we should be reading this right to left or left to right. The gene could be running left to right or right to left. There's not really this, it's not like reading a book, start to finish. Someone needs to mop. Sorry for those of you listening electronically, I dropped my microphone. Try if I can not do that again. So chromosomes don't just go start to finish. They are linear, but because the DNA is running in two directions, they don't just get read from one tip all the way down to the other tip. Trying to think of an easy way to show that. There we go. Okay, so this is one way to look at genes. This is just zoomed in on a portion of the fruit fly genome containing the sepia gene, which is what I searched for. And these genes have little arrows attached to them. So sepia is running left to right. This GST02 is running right to left. And I suppose it's just as likely a gene will be running right to left as left to right. So it doesn't really matter. Genes are individual, but they're part of this big old chromosome, so they do their own thing. And they could be heading in either direction. So it's important, I guess, to think about, when you think about DNA, not to take for granted that you're going to read one way or the other. You have to read the signals in the same way that the cell would be reading the signals. So maybe to get creative here, I'm torn between getting creative to show you some variation and keeping things simple to not throw you off. So I think I will keep things simple for now. So let's say we've got this double-stranded piece of DNA, and on there are the parts that we're familiar with. So we've got the signal to begin transcription, the promoter. We've got the signal to stop transcription, the terminator. Also in there, start codon, stop codon. And let's say we've got a little bit of this, that it would be an intron. So the locations of these signals sort of guide this whole process. The promoter, part of the promoter is really the sequence that tells which direction to get things started. So I guess you can kind of imagine what this double-stranded molecule would look like. If we had a promoter, let's say its promoter sequence is 5 prime TA, TA, G, G, A, T, A, something like that. Let's say that's our promoter sequence. We typically write one strand of the DNA sequence because we're smart people and we can figure out the other strand if we needed to. But the characteristic, the molecular structure that this promoter sequence makes, that kind of recruits the enzymes and sends them off in one direction or the other. So you could have a piece of DNA that goes TA, TA, G, G, A, T, A. And if it appeared this way as the promoter, that would cause transcription to happen towards the right, right? So the sequence we were looking for is running 5 prime to 3 prime on the upper strand. Here's a question. Which one of those strands would be the coding strand? (50-50) chance. Top strand, bottom strand, top strand, bottom strand. How do we know? Somebody knows. Somebody's bold and wants to take a stab at it.
The top strand?
Top strand. All right. Flip a coin. Top strand is correct. So the coding strand ends up running in the same direction as the RNA molecule will get made, right? So direction, I'm talking about the 5' to 3' direction. The RNA molecule is made in the same direction as the coding strand. And the arrows help out here. So I told you that we are reading left to right because of the sequence of the promoter. Everything's going to happen toward the right direction. So sort of toward that direction. And also, as things happen, they'll happen sooner, near to the promoter, and later, farther away from the promoter. So it's really just like reading a book at this point. Once we've established the direction, we're just going to read that DNA sequence and turn it into the RNA sequence. So coding makes that the complementary strand to non-coding. And another way to think about these is one of these is the template, and the non-template is the other one. So I'm trying not to gum up the image too much. Maybe in a little while I'll show you the DNAs and then the RNA that would be made from it. All right, let's say alternatively, we have a promoter that looks like, I can do this, A-T-A-G-G-A-T-A-T. So let's say we had a promoter that looked like that. It has the same DNA sequence we saw above, right? You see it buried in there? Except now it's running right to left, but it's running right to left on a strand that is still 5' to 3'. So there is a strand that is the arrow running this way with that same sequence, just happens to be going the other way. So in this case, the location of the promoter on a sequence like that would have transcription starting that way. And in this case, the bottom strand is coding strand. Top strand is the non-coding. So I'd encourage you, as much as you get a chance when you're preparing for the exam and making sure you did the assignment correctly, just prepare yourself to think about the relationships between these two strands of DNA that are complementary and anti-parallel, and how they relate to the RNA strand that's going to be made. So let's maybe do that. So let's say we have switch things up a little bit. Give ourselves some more room. Okay. So let's say we are transcribing this way. And we'll throw some sequence in here. So if we were transcribing, if this part of the DNA molecule was actually becoming RNA, what would it look like? Would the 5' end be to the left or to the right? Too many 50-50 questions. 5' left or 5' right? Noah.
Left.
Left. Right. No. Somebody's cruel joke on the English language. Right as a direction also means correct. Correct. 5' end is toward the left. So, how'd you figure that one out? Lucky guess?
Well, before when we were looking at which strand was the coding strand, it was the one that had the 2 G's in it rather than the 2 C's in it. That's what I looked at there.
All right. Well, I made up a new sequence here, so 2 G's would not. All right. So I guess we'll chalk that up to lucky guess, maybe. Okay. So I drew you an arrow for the direction of transcription. Again, I think you may be overthinking this sometimes. If the direction of transcription proceeds left to right, then the molecule that's made proceeds left to right, and left to right is 5' to 3', right? Okay. So if those are our strands of DNA, what would be the first nucleotide that shows up in the RNA molecule as it is there?
C.
I got to vote for C. Any other votes?
G.
I got to vote for G. So we're all 4 nucleotides.
I think it's G.
All right, 2 votes for G. Thanks.
Would it be C?
2 votes for C.
T.
T. F. Oh my goodness. All right, maybe I think about DNA more than you do. All right, I'm trying to lay this out in a way that's clear, and I think there's just a tendency or a drive to think through this too many ways. All right, so we have our RNA molecule with an arrow pointing this way. We have our DNA molecule. It has an arrow pointing that way and an arrow pointing the other way. If we are making our RNA molecule in this direction, then it is going to have a sequence that looks like the sequence on the piece, the DNA strand, that's running in the same direction. So that would make that strand the coding strand, and this one the non-coding strand. So these nucleotides are put a wiggle same. So the sequence is the same as the coding strand with that exception of changing our T's into U's. And it's complementary complementary to the non-coding strand. Just make this go. Oh, nice. It works.
Question.
Yes.
So when it's originally transcribing, it uses the non-coding strand to transcribe it, which would in turn make it the same as the coding strand.
Yes.
Okay.
Right, so that's another layer of thinking on this. Jenna pointed out that the process of transcription uses the template strand, which is the same strand we're calling the non-coding strand. So I could do this again, where we split up the DNA molecule. And so it's got a C, G, G, T, A, C, T, A, T. And that would be complementary to GCC ATGATA. So in the double-stranded DNA molecule, these are complementary. So I just expanded out what's drawn above. But if we are making the RNA molecule, then the RNA molecule, if it runs in this direction, then it's going to be complementary to the template strand. So what I'm writing here, this is more like what the molecular process would do. It compares the template strand of DNA, and it uses that to make the RNA. And that's going to happen through this whole region that gets transcribed. So I might have put more emphasis this semester on looking at the coding strand. But also important to know if you had the template strand, also to know how to turn that into RNA. All right, how are we doing? So I might have gotten, I don't know, we'll see how useful this is. I thought I'll probably just go back to the assignment and we can do some of that together. I just chose a gene. This is a human gene called Oco2. This is involved in eye color pigmentation. And it's awful big. But this little schematic here shows us a direction going left to right. Big introns, these areas that are just the horizontal line, are the introns, and then these little bars are the exons. So it's like massive intron to exon ratio. Not entirely sure this will be helpful. We'll see what happens. So you can pull up the DNA sequence for Oco2, and it's a big one. So this whole gene region is 380,000 nucleotides, one gene. Eukaryotes are not known for doing things terribly efficiently. So big old introns. What it says on here is the location of the mRNA. So it says that the gene is located between position 1 and position 380,335. It says that the mRNA starts at 16,325. So we could zoom over. And I'm going to use my, I have a little text editor, and it tells me what column I'm in. So this is the number of nucleotides, 6,000 nucleotides. Keep heading over, 16,000 nucleotides. Oh, we got 12,000, 14,000, 15. All right. So 16206, 16400. Thank you. Sorry, I made a big file. All right, 16325. This is the beginning of the what would get transcribed. So again, I guess I wanted to show you this. When we think about DNA sequences, we typically think about the coding strand. So in Introduction to Genetics, you learn about the coding and the non-coding strands. But once you're comfortable thinking about that professionally, you would just learn about the coding strand and just keep the non-coding strand in the back of your mind. So if this was the start of what gets turned into a RNA molecule, then somewhere upstream of that would be the promoter region. And I was looking for, and I don't see, so I don't know offhand what the promoter sequences would be. Somebody knows, I didn't look it up, but there'd be some part of that that is the promoter sequence that helps these transcription factors and RNA polymerase get attached. That would happen upstream, which is to the left of this location. So back to our diagram here, there would be a promoter. And I should maybe put a little arrow in here. So somewhere around the location of the promoter would be where transcription starts. So it's downstream from some of the important promoter elements, and it's upstream of the start codon. So if we go back to our sequence that I don't have highlighted anymore. Nope. Do have highlighted. Now we're highlighted. Do we start with a start codon? What was our start codon? It's a good one to know.
You say the start codon?
The start codon, yeah.
It's AUG,
AUG,
right?
right. So it'd be an AUG on the RNA, or if we're looking at the DNA, it would be an ATG. And our RNA molecule did not start with a start codon. So that's the one thing that people typically get hung up on. They see start and they think, okay, that starts everything. But there's a different start to making transcription happen than the start to making translation happen. And offhand, I don't see. I'm going to see, maybe I can search for one of those.
There's one right on the right side.
Oh, you found one.
Right.
Oh, there you go.
Those two.
Oh, yeah.
All right, there you go. I do like this program. Once you highlight something, it shows you other instances of that thing. So let's see. mRNA, we're going to skip down. So the mRNA has these codes that say basically this is a set of nucleotides, and then the comma separates out some. The commas are going to separate out where the introns have been removed. And I don't think I can do this simply, so we'll just kind of keep in mind that there are introns in there. This region called the CDS is the code determining sequence, and that's the part of the RNA that actually has codons for the amino acid sequence. And it tells us we're going to find the first one at 16414. So it wasn't 16400, but maybe it's this one. So it tells us that's going to be our start codon. I usually give it to you more simply, and we just do the first ATG we can find. But it's not always the case. Just kind of file that and forget it. Also kind of a weird thing here. It says 16414 to 16416. That's the single codon. And then it says after that, we're going to skip ahead to 17448. Don't worry, I have a plan. All right, 17448. Okay. Let me just copy a bunch of this. So this would be, for whatever reason, that first exon was just the single codon. But then the next exon is about 250 nucleotides long, and I'll just copy the beginning of that. And we can paste it into the translate tool. We'll see how this goes. Translate. And what I want to do with this is compare it to what GenBank says is the amino acid sequence. So the first amino acid was methionine. That was for that one little bit there. And then it goes REDWQW, REDWQW, SMHLE. All right. So we're able to see the amino acid sequence by translating the right part of that nucleotide sequence. Impressive or not so impressive? All right. I feel like maybe looking at 380,000 nucleotides is probably not helpful. So if you weren't following along with the nucleotides, basically we took this really long piece of DNA, and it told us that part of that became the final mRNA. So they kind of skipped this RNA processing step. But there was the start of the RNA molecule. And that RNA molecule would be broken up into introns and exons, and the final mRNA should contain only the exons. So the DNA sequence had instructions for introns, instructions for exons, and it requires stitching all those exons together before you get a final mRNA that you can translate. So if we looked at our DNA sequence for the Oco gene, you could get really familiar with this, get familiar with the ins and outs. You could probably find literature that tells you where the promoter is, what it looks like, what transcription factors interact with that and cause transcription to happen, information about the introns and exons. And then ultimately, sometimes you can just get the single nucleotide sequence, but they're telling us at least the amino acid sequence of the final thing that gets made. My goodness, was that helpful or not? Too late. We put in too much time. Okay. So I had questions mostly on questions 8, 9, and 10. As an electronic file, you should be able to search through here and find most of the answers you're looking for. So question 2 here indicates the transcription factor sequence, TTA-CC, and then it has these ambiguous nucleotides A or T, A or T, A or T, A or T. And that seems to be similar for both of these. So these are a lot of A or T's. These are a lot of A or T's. And then there's this GG sequence that's not found in this area, but is found here. So this would be, that would be basically the transcription factor attachment site. So we can underline that. Tata box, 25 nucleotides upstream. So you could count down from 50 upstream to 25 upstream. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. I'm honestly never sure whether they count from the left side, the right side, or the middle of these. So somewhere about 50 and then about 25 upstream. So these are a couple elements of the DNA sequence that recruit the transcription factors and RNA polymerase. And then I say this is 25 nucleotides upstream. So we'll count down a little bit more. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 20. 1, 2, 3, 4, 5. So somewhere about here would be the start of transcription. Right? So if I wanted to let's see, the initial RNA molecule, let's say it looked like that. So what do I have to do to turn this coding sequence into the RNA sequence? Do I have to reverse it? Make the nucleotides complementary? Other. What do you think? You're thinking, this is why I asked you. All right, we just spent a whole bunch of time.
Was the DNA sequence or like a template strand or non-template?
Is it template or non-template? So I told you it's the coding strand and the non-template strand. Yeah, so this is one of the most important parts of the description of the assignment. Right, so we saw before the coding strand runs the same direction and has nucleotides that look just like the RNA molecule that's made. So about the same? Right. So this is one, unfortunately on this assignment, if you're not doing it right, you probably end up doing a whole lot of extra work.
I just got it.
Yeah.
Oh dear. You know, it's good to know about some web resources here. So if you ever needed to, I don't know, I'm just going to do a web search of reverse complement.
Yeah, we did that because we thought it was the other way.
bioinformatics.org. Hey, look at that. CGT, CTT. Okay. Doesn't even give us the 5' end, so I'm assuming by convention it's going to start with the 5' end, so CGTAA is complementary to, so CGTAA. So that's complementary running from the 5' direction. I guess that's something worth noting if you ever need a bioinformatics tool to convert your strands. The way to convert, if you have one DNA strand, then the other one we say we would reverse complement it. So that means that we are generating complementary nucleotides and running the backbone in the opposite direction. So again, by convention, things are written 5' to 3'. That's what they did on that online tool. So you would actually read this TAT-GGC to get the reverse complement sequence. But we didn't have to do that. So this was the coding strand. So if you put it on here, it should be in the same direction and should have all the same correct nucleotides. And then, of course, we're going to want to change our, do a find and replace, T's to U's, and we'll see if I can do a few of these. I can't see anything.
You gotta highlight the paragraph.
Ooh, that's a good idea.
Otherwise, you'll change all of your T's to U's.
Yeah, I didn't want to do that. T-U. Still looks like it wants to do that. Replace. I see you. All right, well, let me try one more time. Let's search at the front of this paragraph and do that. If my viewing screen wasn't so gummed up, we'd see this happening. Just today, it hasn't done it for the rest of the semester, but just today it decided to force me into a very small resolution of my screen. Okay. Copied a few too many there. Boop, 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 boop. Okay, all the T's have become U's. Now we have a proper initial RNA. Here we'll do our processed RNA. So I just copied the initial RNA, and we're going to process a couple things. I overlooked the... So I'm going to... I guess I can look for this one. Copy, find, paste. Dun, 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 dun. All right, so I think I put a few more nucleotides in there. Now I've trimmed it according to where it said the transcription stop site is. Keep things a little more accurate there. Also gave you some indication about the intron splice sites. So this one has a couple of uncertain nucleotides, but then a bunch that are certain. We can look for that. I'm going to look for it in my RNA sequence. I'm going to change these to U's. And there it is. And it started with the G. I'm going to underline it for now. That was the 5' splice site. And I had a question from somebody. The splice sites are actually in the intron. So the part of the sequence here that tells splicing to happen also gets taken out. So that was the 5' splice site, and then the 3' end of the intron ends with CAGAT. Search for that with a U. Fortunately, there's only one in there, so that must be it. Underline that. And so I can take it out of there. Okay. All right. So we took out our introns. And one last thing. This is spelled out in more detail in your book. I don't dwell much on the 3' end of genes or transcripts, but if you're interested, that is worth reading about. So a 3' poly A tail would be added to the 3' end downstream of the AAU AAA. So basically, part of this actually gets cut out and replaced with a bunch of A's. And that would be more like the final version of our RNA molecule. Questions up to this point?
No, makes sense now.
Well, that's good.
I have one question. So
Yeah.
if you have more than one start codon, does that matter as far as like translation goes, or is it just gonna start at like the first one and then if that makes sense? I don't know.
Sure. The question is if you have more than one start codon. So it's not, you have only so many combinations of nucleotides. It's not uncommon to have an errant start codon. This sequence would be full of them. I said full of them. Can't write this stuff. Okay. So I can scroll through here, and they're going to be all over the place. So there's one start codon that is in the right position. So it depends on where the ribosome gets attached, but it's going to be far towards the 5' end of the final RNA molecule is the first start codon. And in eukaryotes, that's from that point on, that's where the reading frame is for translation. So if it was here, then that would cause next time I don't paste 380,000 letters. That would cause all these other letters to get read as codons. And so if there is another ATG, if it's in the reading frame, ATG will just bring in a methionine. So it'll be like a methionine after the first methionine. If it's outside of the reading frame, then it won't get read as a codon. And it'll just be like you can have some of the mutation content talks about reading frames, but if you had like this same sequence and you were reading it, you could read it in 3 different reading frames. So the same sequence to start with, only one of those has ATG in the reading frame. So yeah, if you're just sort of scanning through a sequence looking for ATGs, the version I give you will be just the first one you could find after you've got your final mRNA molecule.
So it would matter more so if there was like stop codons that would affect.
Yeah, stop codons would be a big deal.
Okay.
So yeah, same thing. The stop codon is really the first stop codon that exists in a reading frame.
Okay.
And maybe we'll get a chance to talk about that. Okay.
What if you've got more stop codons than you do start codons?
It's not a relative amount. It's a location and reading frame issue. So, all right, let's see if we can maybe work on it with the sequence we have here. So I look for a start codon. There's a lot of AUGs in there, and we would just treat the first one as the start codon. We could look for a let's see if it lets me do this. No, I don't want to do that yet. We could look for our stop codons. There's a whole bunch of stop codons potentially in there, but I don't know which ones are part of the reading frame. So UAA is one of our stop codons. UGA is a stop codon. And is it UAG? Yeah, UAG. So there's actually a lot of potential stop codons in there. But for the gene to make the proper protein, then it needs that whole sequence from the start codon all the way to the proper stop codon without any wrong stop codons in there. Okay, so we could just take this and try another translate there. So I didn't paste it with the start codon that we know about, and that might actually be helpful for us to look at the options. I kind of like this website because it gives you options, right? So list things in different reading frames in different directions. So we should ignore the 3' to 5' because we know that we didn't make that molecule running 3' to 5', but we could consider all first 3 of these different reading frames. And we want the one that starts earliest, really. So M is here as methionine, as our start codon. And then dashes are indicating the locations of stop codons. So the sequence that we would probably make is the one with the first methionine here. And just from there, for the amino acid sequence, from there until the stop codon that is in the reading frame for that sequence. Okay, so that was assignment 6. Guess we did do the whole thing. Okay, so I would like to get through the mutation content before we draw the line on what shows up on the exam. You probably had this before, and it's not too complicated, I don't think. So we got up to this point last time talking about where it might be important, where mutations might have bigger or less effect. So we talked about the difference between a mutation, which is simply a change in the DNA sequence, and the effect on the protein. And a lot of the take-home from, like, what is the effect? The take-home is that it depends. And this is where, where the scientists make their money, but I don't know if there's a whole lot of money to be made in this. Get famous for finding out just what ways you can break a protein and have it still work, or which ways you can break a protein and have it really get messed up all of a sudden. And you can really have single nucleotide changes that do either of those, depending on where they happen. So I mentioned, these are just some examples of mutations that can happen. The right-hand side, these are, on the top, these are the standard or usual base pairings, and then these are some base pairings that are, they're basically wrong, but they're a little less wrong than other types of pairings. So C with A, they can make this bond. It's not the best bond, but it might happen as a mistake during DNA copying. G with T is the wrong bond, but it might be okay. Might be missed as a mistake. And that can cause changes to happen in the protein. So let's see what kinds of changes we might encounter. I think I'll probably just walk you through these. Left a lot of questions on your shoulders today. So we'll just sort of start in with kinds of mutations, single nucleotide changes that could potentially change the sequence of amino acids and therefore potentially the function of the protein. So we've got here an RNA sequence, a truncated short RNA sequence, and focusing on this UGC codon, there are 3 possible single nucleotide changes shown. One of these has no effect on the protein. So UGC, I guess the question is, how do you know if it does not affect the amino acid sequence? You should probably find out what it normally codes for. So UGC over here to cysteine, right? So one of those changes would not change the amino acid, would still make a cysteine. And that's that change here. If the C changes to a U. So that would be a mutation. But that mutation still gets you a cysteine at that location. So as far as the instructions for making a protein, you'd still be making the same protein. This is what's called a silent mutation. So really, for all purposes, this would probably have no effect at all for the organism. What if we change it? Which one of these might cause the polypeptide to be shorter? Let's see if I can. Everything's so small. It's not going to work. Not going to work. All right, methionine, GCU, alanine. That was the one. Cysteine, UCC, serine. Okay, so we saw earlier that UGU is also cysteine. If we turn this into a UGA, what happens? So single nucleotide change the C into an A. UGA now encodes a stop codon. And that would cause our sequence to be methionine, alanine, stop. So that one's called a nonsense mutation. It goes from coding for an amino acid to coding for stop. So not only does it cause no amino acid to be put there, it also prevents any other amino acids from getting added. So let's say we have a normal polypeptide sequence. All those circles are the amino acids that make up polypeptide or the protein. If you had a nonsense mutation that caused a stop codon to appear early on in the sequence or halfway through, you would potentially get a much shorter, or at least a substantially shorter, polypeptide sequence. How do you think that might affect the protein? Let's say you were building a car and you got the wheels in, you got the engine in, and you stopped.
It just wouldn't work.
Might not work. Be cold to drive it anyway. So I'll put here big effect. If you had a stop codon appear, why did I make this so long? Not too far from the end. That might have less effect on the function of the protein. So you build your car, you get everything in, and you left out the windshield wipers. You still got most of a car. Even these simple versions of big effect, little effect, it's hard to gauge. So for one protein, it might be really important, those last 5 amino acids or that last amino acid. They could be very important. Hard to know without being specific about a protein, but I think we can safely say if you chop off 2/3 of your protein, that's going to have a big effect than chopping off the last 2% of your protein. So I like to think about not only what kinds of mutations can happen, but also what effect they might have on the protein and therefore on the phenotype of the individual. All right, good so far? So here's another one. We tried all the other options. If we change the middle nucleotide there, UAC, instead of making a cysteine, now at that position a tyrosine comes in. And that affects only that amino acid and nothing around it, which is kind of handy. It's called a missense mutation. So in the sequence causes the one amino acid at the location where the mutation happens to be a different one. Where's my copy and paste now? All right, so I'll just color it in a different color. What do you suppose the effect would be of that? Change a single amino acid. These are cases where the answer is, it depends. So if we go back to this slide, These are proteins made up of lots and lots and lots of amino acids, but there are 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids that interact at the active site. Changing one of those could cause a pretty big effect. So amino acids interact with their neighbor amino acids. They interact with amino acids across other parts of the protein. They interact with molecules in the active site or some kind of signaling site. And so if you mutate one of those important amino acids, that could ruin the function of the whole protein. If you take one of these helix regions, these coiled up regions, those are typically a little more generic. But if you affected how much these amino acids are hydrophobic or hydrophilic, that could affect protein function. And of course, mutating a protein so that it doesn't work is also a big spectrum, right? So you got broken entirely, can't possibly do its job, or maybe broken a little bit so that it does its job less efficiently. You might get like a minor phenotype. So I guess from lab, some of you worked with the white apricot allele, right? So there was the white mutant that had no pigmentation in the eye, and that was a completely non-functional protein. The white apricot allele gives you a protein that functions a little bit, and that apricot eye color allowed some of the pigment molecules to get through. So this is definitely a whole field of biology devoted to looking at molecules and how they're made, how they function, what the mutants look like. Okay, so I kind of jumped ahead and we covered a lot of this. So missense mutation, single change. Nonsense mutation makes the protein shorter. And silent mutation, not really an effect. I'm going to skip over that one. And jump ahead. I guess I'll just introduce this one. We're running out of time. What we've looked at so far are single nucleotide changes. And another possibility is that more than one nucleotide gets added or removed. And that messes up a whole lot. It's called a frame shift, and the frame here refers to the reading frame. And I think my best example is if you just use English words, 3-letter words. So I got a sentence of 3-letter words. The dog was big and red. Let's say we insert a letter in there. Boy, does it stop making sense. So if this was a gene and we did a frame shift, then those codons don't code for the same amino acids anymore, and they've more or less randomized into other kinds of amino acids. They're not making what they're supposed to, and this part of the sequence would just be pretty well ruined for the function of that protein. So I added a letter. Let's take a letter out. Take one out there. B-I-G-A-T-R. Yep. So same thing here. We're not making letters anymore, we're making codons, but for different amino acids. And that sequence of amino acids is really the basis for the function of the protein. And if it's not the way it should be or evolved to be, similar to what we saw with the, we'll pick back up with this next time, but similar to what we saw before with the nonsense mutation, rather than taking out an amino acid or part of the sequence, it becomes a different amino acid sequence from that point forward in a frameshift mutation. So that's it for our time. Come with questions next time, and we'll do some review if you want to. We'll finish up the mutation content. There's not much more left. Have a good weekend. Is it Thursday already? Wow.